Alright, we are currently on the topic of properties of monosaccharides. Now we're going to look at two different reactions of monosaccharides. And so in this chapter, we're going to identify the products of both the oxidation or the reduction, hydrogen gas, of these monosaccharides and determine if a carbohydrate is, for example, a reducing sugar. And we'll come back to the reducing sugar because I think there's a bit of a confusion around reducing sugar and then reducing sugars. They're two different things, so we'll talk about that. What I'm showing you here is actually that reduction. So you can see here that a sugar like D-glucose can actually be reduced. Remember that an aldehyde reduces to a primary alcohol, and a ketone would reduce into a secondary alcohol. All that means is that we're going to increase the number of bonding to H and decrease the number of bonds to O. So you should see that with this carbon here. It has an extra hydrogen and one less bond to O. So we'll talk about what the products of those are. Well, actually, I guess I'm showing you a product here. So you can see here that D-glucose changes its name to be D-glucitol. And so this is actually a sugar alcohol. We call them sugar alcohols. D-sorbitol, which is the other name for D-glucitol, or like D-xylitol, D-xylose. D-xylitol would come from D-xylose, and D-mannitol would come from D-mannose. So we'll change the ending "-ose" to "-itol". But a lot of these sugar alcohols are used as sweeteners in like sugar-free products and diet drinks and in low sugar diets and that kind of stuff as a sugar substitute. You can see here sorbitol in this pack of, I assume, sugar-free gum. So the oxidation is what we'll talk about first. Monosaccharides, they're mostly found in the cyclic form. But remember, there's that mutorotation that happens. The cyclic form opens and closes, opens and closes. Well, when it's open, it does have that aldehyde group present. And that aldehyde group present with an adjacent hydroxyl, we talked about in the aldehyde chapter, can be receptive to a particular kind of oxidizing agent called the Benedict solution. And a Benedict solution uses the copper 2+ ion, and it gets reduced into a copper 1+ ion, and in that process oxidizes the aldehyde into a carboxylic acid. So aldehyde into carboxylic acid. Remember, ketones can't oxidize further. So this was a test that we could use to determine if our substance was an aldehyde versus a ketone, or in this case an aldose versus a ketose. But we can also use it to detect the presence of glucose, and this is often used in tests medically to see if someone, for example, we can test the urine and see if there's sugar present, and if there is, that can often be associated with kidney damage or some sort of kidney problem. So oxidation of a aldose will yield a carboxylic acid. Now those are called sugar acids. I don't think I have another picture of it. So sugar acids are produced from that oxidation. It doesn't have to be the Benedict solution. We name those usually by changing the OSE ending from the sugar to a onic acid ending. So for example, mannose would become mannoic acid. Fructose, fructoic acid. Galactose, galactoic acid. Now fructose, I misspoke. Fructose can't oxidize into an acid because fructose is a ketone. So forget that I said that. But galactose definitely could. It could do that. Okay. Now, carbohydrates that oxidize are called reducing sugars. So let me just show you this one more time. So let's say that I have glucose and I have copper 2+. We react it, we get copper 1+, and gluconic acid. So glucose here was oxidized. I don't think we're confused about that at all. Copper was reduced. Now, for copper to be reduced, it needed a reducing agent, something that could reduce copper. So glucose was the reducing agent. And likewise, for glucose to be oxidized, it needed an oxidizing agent. That's what the copper was, remember? And we usually show that as an O, oxidizing agent. That's the thing that gets reduced. So when we say a sugar is a reducing sugar, it means that it has the ability to be oxidized. So therefore, only aldoses are reducing sugars. Now, as I say that, fructose actually does often yield a positive Benedict solution test or a positive result with the Benedict solution. And the reason for that is, even though fructose is a ketohexose, and I like how it says here usually can't be oxidized. Well, they can't be oxidized, but fructose, which normally shouldn't be able to be oxidized, can actually undergo a rearrangement where the carbons on number 1 and number 2 switch their structures. Let me show you here. So here's fructose, ketose, a rearrangement happens and we end up with an aldose. And since the rest of the molecules are the same, that effectively converts fructose into glucose. And glucose can be oxidized and is a reducing sugar. So often, fructose is referred to as also a reducing sugar, but that's only because in water this rearrangement happens and it effectively becomes glucose, and glucose is the thing that's the reducing sugar. So let's just say fructose is not a reducing sugar, but fructose can turn into glucose, and glucose is a reducing sugar. So we can also reduce monosaccharides. The reduction, of course, remember, yields alcohols, primary alcohols for ketones, for aldoses, and secondary alcohols for ketoses. Now, any kind of reducing agent can be used. Typically, hydrogen gas with a platinum or palladium or nickel catalyst is used. But remember, your homework also shows sodium borohydrate can be a reducing agent as well. We get the alcohols. And again, we name these as itols or itols. I don't know if I have a picture. Yeah, there we go. So here we have, again, sorbitol, xylitol, mannitol. These are all sugar alcohols. And again, they are used as sweeteners because they often still provide some sort of similarity to the sugar molecule on our tongues, which our tongues recognize as sweetness, but often are not metabolizable in the same way that glucose is, and so they pass through our system often or sometimes undigested, and so they don't have any caloric value. There are others that can be digested to some extent, so they have less caloric content. So they're just therefore low sugar, or sugar-free technically, because they're not saccharides anymore, they're sugar alcohols, but also, they still kind of have some energy in them. Okay, quick learning check. Here we have D-mannose. Your objective is to draw the product of the reduction. So just remember, it becomes an alcohol. Since that carbon that the alcohol forms on is no longer chiral, or isn't chiral, you just represent it the same way that we would show a non-chiral carbon. So let's get into the disaccharides now. So disaccharides, like lactose here, are containing multiple monosaccharides. They could be the same, they could be different. In this particular example, lactose here is made up of 2 different monosaccharides. It has a galactose and it has a glucose, and you can see here that there's actually some more specificity. There's a beta galactose. Of course, all of the ones that we look at today will be D versions instead of L, or D enantiomers instead of L, but Beta galactose is particularly important instead of alpha galactose. Remember, alpha would have this bond going down instead of going up. So beta galactose and then alpha glucose. Now, alpha glucose or beta glucose actually aren't going to make any difference here, because remember, that's just the position of this OH, and since this part of the disaccharide isn't tied up in an acetal bond, since this part of it still here is a hemi-acetal, it's a little unstable, and it can still open and close, open and close. Therefore, it can still be oxidized, it can still form another acetal reaction, but it can also mutorotate. So let me write all those things. It can, I already forgot what I said. It can mutorotate. That means it can turn into beta and alpha back and forth because it opens and closes. It can form an acetal, and I guess open and close was the other thing I said. It can still open and close. So the hemiacetal bond there is still hydrolyzable. It can still hydrolyze. I think that's what I said. So that these are open and closing is still sort of related to the mutorotation. The formation of the acetal bond will stabilize it. Oh, I remember what I said now. It can be oxidized, meaning it can be a reducing sugar. Reducing agent, I wrote. Reducing sugar is what I wanted to write. RS, reducing sugar. Okay, when we have bonds between two sugars, we often refer to that acetal bond as a glycosidic bond or a glycosidic linkage. And we like to specify the glycosidic linkage with some more information. For example, what orientation the leading bond molecule was in, whether that's alpha or beta, and also what carbons are being attached. So you can see here for this glycosidic bond in lactose, and what's shown here is the alpha lactose, simply because the OH group is down on the position of the carbon that still has an OH. But you can see here that the bond is called beta 1,4. Beta because of the up position of the previous hydroxyl, but also because 1,4 is the two carbons that we're linking together. So let's take a look at some more examples. I was going to detail some of the other disaccharides. So we have maltose. Maltose is made up of just alpha D-glucose with it doesn't matter what it is, alpha or beta D-glucose. And of course this would lead to alpha maltose or beta maltose, but maltose is simply made up of glucose. That's a disaccharide. We're going to see some polysaccharides as well that are only made of glucose. Then we get to lactose, which we've already looked at. Galactose is a beta D-galactose with an alpha or beta D-glucose. And then we have sucrose, sucrose. And sucrose is going to could be composed of let me think. It's an alpha glucose and a beta fructose. So beta D fructose and alpha D glucose. And you're going to see here, in this particular situation, it has to be an alpha. We'll come back to that one when we take a look at it. Let's look at maltose first. So maltose is a disaccharide known as malt sugar. It comes in, found in grains and that sort of thing. It's 2 D-glucoses. It can be obtained by hydrolyzing starch. We'll talk about starch when we look at the polysaccharides, but polysaccharides are just long chains of glucoses, so if you start chopping those into little chunks of 2, those will be maltoses. Used in cereals, candies, and in brewing. I often make beer that has malted barley in it. And then we have alpha and beta forms because the second sugar, as you can see here, the second sugar still has a hemiacetal, so it can open and close, therefore muto-rotate back and forth between being an alpha or beta. This one, maltose, is characterized by an alpha 1,4 glycosidic bond. So it does have to be an alpha glucose in the beginning so that the OH is down, so that we get this particular arrangement in space. We have enzymes in our body that are able to break that bond. Water would break this bond normally, but in metabolic processes, when you're trying to get energy from these compounds, you want to be able to break them at will. So we have enzymes that specifically find sugars and cut them in half. If the bond isn't right, we can't digest that, and we'll talk about that a little bit when we look at lactose here in a second. So yeah, that's maltose. So let's take a look at lactose. So what you need to know are the sugars involved in making the disaccharide and the bond that would be part of that disaccharide, as well as if you were to be drawing a disaccharide. And here, think about this. I can give you any two sugars. I can give you two sugars and say link them with a beta 1,6 linkage or a alpha 1,2 linkage, and you would just connect them knowing what carbons are connecting and what direction the OH is going to be, or the O bond, based on alpha and beta. I could even give you two Fisher projections and say draw these as Hayworth structures and then link them with an alpha 1,4 or a beta 1,4 link. So you can do that with any sugar. It doesn't even have to be glucose, galactose, or fructose. I could even show you a disaccharide you've never seen before and ask you to tell me what the linkage would be. And all you have to do is look at it and realize that the carbon off of the oxygen is always the number 1. Whatever other numbering system is going to be based on where that number 1 is, so you could identify that this was the 4. And then just the position of the bond, whether it's down or up, is going to be the alpha or beta. Let's look at another example. Let's look at lactose. So lactose is the disaccharide of beta D-galactose and an alpha or a beta glucose. This is again found in milk products and dairy products. About 6 to 8% of human milk is lactose. About 4 to 5% of cow's milk is lactose. Here's the bonds. You can see here the up beta OH, and then the in this case it's an alpha glucose, but it could be a beta glucose. It's the 1 and 4 that are going to link together. If you want to talk about the acetal formation, it's the hemiacetal pairing with an alcohol. Remember, hemiacetal plus alcohol equals acetal. It just happens to be the alcohol group on the number 4 carbon. So as they link together, we represent it this way with the bond up. I don't remember if on maltose the bond was up. No, the bond was down. So this is what an alpha bond looks like. Right? It's down. And a beta bond is up. Again, beta 1,4 linking the number 1 and the number 4 together. And this is alpha lactose because the OH is down over here. When these form, they release a water molecule. I think I forgot to mention that in the previous example. When maltose forms, it liberates a water molecule. Dehydration synthesis, we call it. And you can see that here. It's highlighted in pink. So this little pink box has the OH and the H that are the water. So that's lactose. Now, this one again has a anomeric carbon that can flip open, and the ring can open and close. It can be oxidized. So lactose is a reducing sugar, just like maltose. But now let's look at sucrose, table sugar. Now, first thing let's take a look at is the bond, and I think this is written a little funny. This is an alpha beta 1, 2. That means that it's alpha on the first sugar, beta on the second sugar, and that it's the number 1 and the number 2 carbons. Well, when we take a look at the number 1 and 2 carbon of this particular pair, for glucose, that's our normal number 1 carbon, and alpha means that it needs to be down. But then we see that on beta, because, on fructose, this is that 5 membered ring. This is the, the, the pentose sugar. It forms not a pentose sugar, it's still a hexose sugar. I'm trying to remember now what the word is for the 5 carbon rings, and I can't remember what it's called now. Anyways, it's a 5-membered ring with 6 carbons. The OH group in this case is up. This is why it's beta. And it's the number 2 carbon here that is the anomeric carbon. So this is, instead of it bonding to the number 4 carbon like we've been seeing in the last 2 examples, these are both anomeric carbons that are forming. So this hemiacetal, or these 2 hemiacetals are forming together, making one acetal, where the last example, there was always a reaction and then there was still a hemiacetal ready to grow, like to react again. You could foreseeably take each of those disaccharides, maltose and lactose, and still add more sugars to the growing kind of chain, make a growing chain by adding to the free end. But this molecule, both of the free ends, both the sides of the ring that could open and close, are now sealed. This bond over here is stable. It's an acetal bond for both sugars. This does not open and close. This does not oxidize. This does not mutorotate. This is not a reducing sugar. So, yeah. I forgot to mention, going back here to lactose. Lactose is the beta-1,4 glycosidic bond. If you're lactose intolerant, it's because you don't possess an enzyme that's capable of breaking down that bond. And sometimes people that are, not necessarily do they have the enzyme, but they have the right gut bacteria that are able to break down lactose into these 2 sugars, which your body does have the ability to digest or to metabolize. But to free up those sugars, we need to get it out of this disaccharide, which some people are unable to do. Okay, sucrose. We talked about some of these things already. It can't be oxidized. It doesn't react with the Benedicts. The sugar that we use, again, we called it table sugar here. This is sucrose. We put it in our cereal or coffee or tea. Most of it comes from either sugar cane or beets, sugar beets. Okay, quick learning check. Go ahead and identify the monosaccharides that are in each of the following. So out of these 3 options, which 2 are in lactose, which ones are in maltose, which ones are in sucrose. So pause it and give it a shot. All right, hopefully, I guess here it could be the beta as well. We could also have a beta as the second one. Probably doesn't happen very often, or even if it does, it opens and closes, probably stays as a beta more often when it's closed. And then for sucrose, these have to be an alpha and a beta. Excuse me. All right, another learning check. This is melibiose. This is actually sweeter than sucrose, so this is a disaccharide as well. Notice that it's a little bit different. So here, you are going to look at these 2 units and tell me what are the monosaccharides in here. Pay attention to the positions of the OHs. Tell me what kind of linkage this is, and what we would call it, and then identify this whole thing as being either alpha or beta. So go ahead and pause this and give it a shot. All right, so that's an alpha D-glucose and an alpha D-galactose. There's an alpha 1,6 glycosidic bond, and because the position of the OH was down at the end, it was alpha melibiose. All right, last but not least, let's talk about some polysaccharides. So polysaccharides are formed, by the way, I'm showing here a picture of cellulose. You can see that here. It comes from plants. It's a main component and structural component of plants. But we'll get into some of the more interesting things about cellulose when we can compare it to some of the other polysaccharides. Now we're going to focus on polysaccharides of glucose. There are other polysaccharides of other sugars, but this is the monosaccharide that we're going to pay attention to. And the reason is because there's quite a few polysaccharides of interest that are all just made up of glucose. So a polysaccharide forms when we have multiple monosaccharides joined together. Amylose and amylopectin are both components of starch. They're no different in structure, or well, they're no different in their chemical makeup. They are slightly different in their structures than glycogen, which is what we use. This is like animal starch, if we called it that. Animal sugar. Starch is plant sugar. And then, again, amylose, amylopectin, glycogen, and even cellulose, which is also a plant structure, are all D-glucoses. And so the way that they differ is in their branching and in their glycosidic bonds. So the goal in the end, in this chapter or this section is to be able to identify the differences in their bonds and to know the differences in their structures. Okay, so first off, let's talk about starch. Starch is composed of amylose and amylopectin, and different plants have different percentages of these two types of storage for glucose. Plants want to store glucose for various reasons, for building, they metabolize the same way we do. They need to burn fuel to build their bodies, and so they store energy. Now, the energy demand of a plant may vary. Some plants, like a Venus flytrap, for example, that needs to be able to slam its little mouth closed or something, might have a higher need, and so it may have more amylopectin than amylose, and you'll see why amylopectin is a little bit better for energy here in a second. But this is what amylose looks like. It's a long chain of alpha D-glucoses. You can see here the alpha down for all of these bonds. So that's why it's the alpha, and it's from 1 to 4. So alpha 1, 4. And this just goes on and on. It's an unbranched chain of glucoses. Now that differs from amylopectin because amylopectin has branches. So about, amylose, which is about 20% of starch, roughly, has about 250 to 4,000 glucose units connected by alpha 1,4 glycosidic bonds, and they kind of coil in a helical fashion. The branched chain just has branches. That's kind of what it is. And by putting branches on there, you can actually fit more glucose. And one of the benefits for having more glucose is that starches can hydrolyze easily in water. Well, in water and acid in the lab. But in our bodies, we have enzymes that would do this. But in water, they can hydrolyze into smaller bits, and actually I have that here. Amylose and amylopectin can be hydrolyzed into dextrins. If you keep hydrolyzing, we get maltose. Keep going, we get glucose units. But it's the ends where we can hydrolyze. It's where our open chains or where our reacted bits are. So let's say this is the end sugar here. We can come in here and cut this and then eat this sugar, and then we come in here and cut this one off and eat it, and then cut this one off and eat it. The more branches we have, the more ends you have, if you can picture that. If I'm just a straight chain containing organism, I have to cut these sugars off one at a time. But if I have all these branches, then now I have 3 ends that I can be cutting sugars off from. My enzymes could be coming from all 3 ends. The more branches you have, the more available sugars you have. So that brings me to humans. So humans use glycogen, or we call it glycogen. It's basically just amylopectin. It's a polymer of glucose that we store in the liver and the muscle. It can be hydrolyzed in our cells that maintains, levels of glucose that we need. Of course, we can burn through it all in about a day. It's just more highly branched than amylopectin. So it also has, I skipped over showing you the other bond, the important bond here. So it's still connected by alpha 1,4 bonds between glucoses, but where the branch happens, it's actually connected from the alpha number 1 to the number 6 of the branching point carbon. So another number 6 carbon here could have another branch, and another number 6 carbon can have another branch. So alpha 1,6 and alpha 1,4 are present in galactose. I'm sorry, galactose. They're present in glycogen as well as amylopectin. Glycogen, I don't know if I have a picture of glycogen. Glycogen is exactly the same. Alpha 1,4, alpha 1,6. It has branches that occur every 10 to 15 units as opposed to maybe every 20 to 25 units. So just more branch, more packed together glucose, more energy. Now, cellulose differs from all of these because cellulose contains beta D-glucose. And simply having beta D-glucose leads to this bond here being a beta 1,4 bond instead of an alpha 1,4 bond. Well, this, well, a couple of things happen from this. One, it doesn't form as many hydrogen bonds with water, so it's actually less soluble. It doesn't have the same sort of helical structure. It has a more rigid structure to the cell that it provides to the cell wall, so it's useful for providing some sort of cell strength. We find it in wood and fibers. This is what makes them tough. It's more resistant to hydrolysis than regular starch. It can't be digested by humans because we don't have the enzyme to break down beta 1,4 glycosidic bonds. And so essentially, cellulose is a really good fiber. It doesn't dissolve in water, and we can't digest it. There are organisms that can digest this because they have enzymes within their gut bacteria that can break down these bonds, freeing up the glucoses to then be consumed. And once you break this apart, this beta D-glucose can instantly become an alpha D-glucose. As long as it's a D-glucose, we can use it. All right, learning check. Identify the polysaccharide and the types of glycosidic bonds in each of the following. So A and B. Pause it, give it a shot. A is cotton and B is a potato. I don't know what's going on with my arrows here. So A was cellulose, it only has beta 1,4 bonds. B was a starch, so it contains amylose, which is alpha 1,4, and amylopectin, which is alpha 1,4, alpha 1,6. There's a couple of other disaccharides that I want to talk about real quick. Glycosaminoglycans are a type of unbranched polysaccharide that consists of repeating disaccharide units. So I guess it's a polysaccharide. But the disaccharide repeating unit is something we haven't looked at yet. We looked at just a repeating monosaccharide unit. The disaccharide unit is composed of two different parts. One is an amino sugar. These are the amino sugars here. So there's alpha D-glucosamine and alpha D-N-acetylglucosamine. And then one molecule of either uronic acid or a galactose molecule. I'm sorry. Yeah, a uronic acid or a galactose molecule, but it looks like uronic acids are just galactose molecules that, this is a galacturonic acid. So this is a galactose that's been oxidized. This is a glucose that's been oxidized, or just a galactose. So it's one of these guys plus either that or one of these guys. And there are obviously different types of glycosaminoglycans. I'm not saying there's only one. There's lots of different ones, and they could be made up of different of these combinations. What are they good for? What do they do? These are very polar, and they're very negatively charged. That means that they can attract water, and they can cushion or lubricate structures in the body. They include keratin sulfate that's found in the cornea of the eye, cartilage, bone, horns of animals. But anyways, they have a role in the body as being lubricants. This is what I'm showing here is actually an unsulfonated keratin deposit that can cause cloudiness and stuff to develop on the cornea. And this could be in relation to problems with glycosaminoglycans or not having enough of those molecules. I'm not really sure actually how the sulfonated part comes into it, but yeah. anyways, here is the concept map that kind of goes over everything here. So monosaccharides, disaccharides, and polysaccharides, know the linkages and know how to draw them, know the definitions of Hayworth structure or Fisher projection, that kind of stuff. And as always, email me or hit me up if you have any questions. And good luck with it, with this material, and good luck with everything else, guys. I will see you in the next video. Take care.